Good morning and thank you for coming. I'm Seung-hyun Lee from Seoul National University and it's such a great pleasure and honor to be here. And I'm here today to talk about the invasive populations of Asian longhorn beetle in Korea. And yeah, let's get started. Maybe I don't need to introduce the ALB to the audience like you, but I prepared, so that's good. As Lindsley mentioned, there is a negative correlation between the range, the number of host plant, and the vitality of host plant. Negative correlation. So the outlier serum-based seeds, which damages many kind of trees, many kind of living trees, can be a serious pest, and the ALB, Asian longhorn beetle, is just the case. ALB is highly polyphagous. They eat more than 36 plant families, and the main host of them are Acer, Papillus, and Salix, and as well as others. And they have been recently highlighted as a serious pest on street trees, and study on this notorious pest was accelerated after they invaded to North America and Europe. And their native habitat is East Asia, namely China and Korea. But the situation of ALB outbreak is quite different in both countries. China went through ALB outbreak. The first outbreak of ALB is first reported in 1985, and because of human act like reforestation or commercial trade, these outbreak area become much larger in 20 years. But in contrast, Korean ALB population has never shown outbreak until recently. In 10-year expedition on Korean serum-based seeds and this literature, and we reviewed museum collections, we conclude that Korean native populations are only found in deep natural forested northeastern part of Korea, and only small populations are found, and they show strong preference to an Acer species, especially Acer mono. Yes, so why? Why there has been no outbreak in Korea? Some say there are some reasons. Natural resistance of tree, high tree diversity, and natural enemies of ALB and narrow host range of them. While others say that there still exists potential of outbreak of ALB in Korea because of recent reforestation of popular species, just like China. And yes, the outbreak really occurred in Korea, but it occurred in a very different way from what they thought. Very recently, we found some strange populations of ALB in Korea in many cities. We found them on Seoul, Incheon, Ulsan, Busan, and Gimhae. As you can see here, all the cities are the harbor cities or its adjacents. And what's important is Busan is #1 port city in Korea, the largest port in Korea, and Incheon is the 3rd largest. So the invasive pest in harbor city, that is very suspicious. If there has been no ALB record in Korea, we could easily conclude that these populations are invasive. But we already have a native populations of them, so this population can be just an expansion from this original native population. So we should check these populations in many aspects. So what about biology of this population? In Incheon, almost every individuals were collected on horse chestnut. And what's interesting is Acer species, the #1 preferred host in Korea, is just near the horse chestnut trees, but all the adults were found on the horse chestnut. And the second preferred host plant in Incheon is Salix species. And in Seoul, all individuals were collected on Acer saccharinum, which is non-native Acer species in Korea. And in Ulsan and Busan, we found a massive populations only on Salix alongside the river. Yes. So their biological trait is quite different from native population, like host preference or population size. And they also show a very different form when comparing to the native population. We examined more than 100 dried specimens of ALB in native populations of Korea. They show really consistent elytral pattern, but you can easily find the wide range of variation on elytral pattern in the newly found population. We also investigated male genitalia, Edagus and Tegman, and also wing venation, but we cannot find any significant difference between two populations. So These newly found populations are very suspicious to be invasive because of these reasons. First, outbreak area is harbor cities and it's adjacent. Second, their unusual biological feature, their host preference and population size. And third, their different appearance, their morphologically different literal pattern. And if we add genetic evidence to these three evidence, we can conclude more strongly that these populations are invasive. But in previous research of ALB in United States and China, they used both mitochondrial DNA and microsatellite. And they conclude that Chinese populations are themselves invasive, which means already mixed genetically, so they cannot pinpoint the origin of invasive populations of America. So if these populations later to be found to be invasive, we could not pinpoint in which part of China this population came from, so we only use mitochondrial DNA analysis to determine whether these populations are invasive or just an expansion of native population. So the molecular works are like following. We use more than 1,500 base pair of mitochondrial DNA from CO1 to tRNA leucine to CO2 from 96 individuals from 8 different locations in Korea is marked in the map. And we also downloaded 196 sequence from GenBank. And here we performed the Bayesian inference tree. And the supporting value is quite reliable. The posterior probability value is all over 0.8 except one here. And here I visualize this tree with the map of Korea. This tree clearly divided into two clades, the upper clade and the lower clades. And all the domestic population belong to the upper clade, and all the invasive population belong to the lower clade. And we can also divide this invasive population into two based on its distribution, namely Incheon, Seoul population, and Busan, Ulsan population. We also performed a CO1 haplotype network analysis, and here's the results. There is a 0.8 to 1.1% difference between native populations and invasive population. And this is the most important part, I think. You can see here that both Busan, Ulsan population and Incheon, Seoul population shares perfectly identical CO1 haplotype, respectively. This means all the individuals collected in this area has the same CO1 sequence, and all the individual from here has the same CO1 sequence. But in contrast, we can find many haplotypes scattered in native population, and this means this is a good molecular evidence of founder effect of invasive population. We think this is the most important part. And lastly, the physical distance does not correlate with the genetic distance. That means the native population and the Incheon, Seoul populations are physically closer, but invasive populations themselves closer genetically. So we found these three facts through molecular work. First, there is a distinct genetic distance between the native populations and invasive population. Second, there was narrow range of genetic variation between invasive populations, and we found many haplotypes in native population. And we thought this is a founder effect of invasive population. And the third, there was no correlation between genetic distance and the physical distance. So let me wrap up. We reviewed 4 things: morphological, biological, and distributional and molecular evidence, and the evidence independently indicates that these populations are invasive. So we conclude that the newly found population, namely Incheon, Seoul population and Busan, Ulsan populations, are invasive. They are not from Korea but from China, and they may be invaded independently at least twice, once in Incheon, Seoul area and once in Busan, Ulsan area. And this is what we got for today, and I would like to thank my supervisor Sung-Wan Lee and my old lab mates and collaborators to make this study happen. And also thank you all to participate and pay attention. Thank you.
have natural enemies come down into those invasive populations? I would think that the separation currently, because the natural enemies are kind of clustered up in the native population, do you think that they'll start to come down and control your invasive populations?
We found this population very recently, so that is not enough for them to come down, maybe. But I believe that in the future, maybe they will come down. Yes.
You said you don't know the origin. You know, it's just from China. Do you have any idea which part of China it came from?
I don't have any idea because all the sequence on the GenBank is not from the natural forest, but from urban area of China. So that is already mixed. So if I want to pinpoint the origin of this invasive population, we should carefully check the ALB in natural forest in China. Maybe. So that will be our future research.
So as I understand it, in China, some of the problem with ALB is that there's a whole lot of populace that's not native to Asia, that's American, that's planted there, and they love it as a host. Does Korea face similar things? Are there plantations of non-native Acer or populace for wood or pulp production that would be a place for these beetles to be happy?
Yes, but as I told that all the populations are now, native populations are inside the forest, the forest.
Right.
So it's.
So they're limited because
Yes.
they're native trees there. Right. And I guess I have another question. Showing your map, it kind of begs the question about ALB in North Korea.
Mm-hmm.
Any knowledge of distribution of that beetle in that country?
I think North Korea is much far than America. When I can.
No, I'm sorry. The beetle's native to China. It's native to South Korea. Presumably, it's native to North Korea.
Yes.
And you don't have. I assume you don't have access to those populations for genetic comparison or any knowledge of invasive ALB there, which may be less likely because of shipping less frequent. I don't know if I'm making any sense, but I'm just curious about it. Kind of. Yeah. So it's hard to have knowledge of ALB in North Korea.
Yes, it's very hard to. So thank you.